the iGEM competition is a competition for designing bacteria with properties and behaviors that they didn't have through Darwinistic evolution. And they are members of the winning team of last year. So how this exactly worked, they will explain in more detail. Eva Brinkman, Nadine Boogaerts, very welcome.
Thank you, Klaas. Good morning, everyone. My name is Nadine Boogaerts, and I will give this talk together with Eva Brinkman next to me. We are, as Klaas said, students from the Technical University of Delft, and we have last year participated in an international student competition that is all about the screws and bolts of the 21st century, the so-called BioBricks. In the next 20 minutes, you will know what these bricks really are. It involves DNA and cells, and you might not be familiar with this, but don't worry, because I will explain everything. The reason why we are here has to do with this competition, and Eva will tell you what project we did for this challenging worldwide project. Of course, if you have questions, we'll be happy to answer them by the end of this presentation. So, before we're going to talk about the screws and bolts of the 21st century, I would like to take you back in time. Yes. It was around 200 BC when the first bolts and screws, the concept of screws and bolts was developed. However, the screws and bolts as we know them today came from the Renaissance. These screws were handmade, and so it was very time-consuming. But a clever man, Jacques Besson, found out how to do automated production. Over time, this process has been improved to production ways we now have today. And because we now have automated production, we can easily build and construct standard parts. So, what can we make using standard parts? Well, all kinds of things, of course, we are all familiar with, like a coffee machine, or a metro station, or a hair dryer. But what if we want to use the same principle for designing nature? How would we do this? Can we use standard material? Well, I don't think this is the right way to do this. So, of course, we need to think of something else. In other words, we need a different type of building block if we want to engineer nature. So, but before I'm going to explain you all how this can be done, we first need to take a look at nature and how it builds itself. I'll show you this video. You see a human here, and I'd like to zoom in. Oh, sorry. Yeah. Let's zoom in to find out what we are made of. We are entering a world of living cells. And each cell contains a nucleus. That's the place where the DNA is stored. I assume you all know that DNA is the material that contains all of our genetic information. So that means that, for example, the information for the color of my eyes is stored in this molecule. Cells are not visible with the human eye. So to give you an idea of how small cells really are, I'm going to zoom into this picture. Please pay attention to the upper left square here to see the reduction in size. We start off with a coffee bean that is about 12 millimeter, and I will stop the video when we reach a bacterial cell that is far more smaller, of course. Hope you can see it. Human egg, a skin cell, and here we approach the E. coli you saw before in the pictures of previous talks. All right. So now we know at what scale and where the DNA is stored, but how then is this information translated into functional proteins, like, for example, the blue pigments in my eyes. This is specified by a segment of DNA we call a gene. The DNA is then translated into RNA, and RNA is translated into the proteins, the molecules that have a function in cells. We can compare this if you imagine, for example, a blueprint that is made for a new metro station, and this is made in Amsterdam. Then a copy is sent to Rotterdam, and in Rotterdam the actual metro station is built and has its function. This might sound easy, but of course this is not the whole story, because the cell also needs to understand where this information starts and ends. And clever as the DNA is, it also encodes regions that tells the cell where to start reading the DNA, how this DNA can be used to build the protein, the actual information for the protein itself, and where the information stops. Knowing all this, we can think of ways how to adjust these regions of code in a way we want. It's very challenging to alter humans by changing their DNA, but bacterial cells that consist of only one cell are far more easy to modify. In this animation, I will show you how this can be done. So we have this sphere-like picture that represents a bacterial cell with this DNA, and somewhere in its DNA it has a gene of my interest. I can take an enzyme that works as biological scissors. And now I cut out the code of interest. This. Of course, it's not a real scissor, but it's far more easy to explain. And I put it in an artificial standard type of DNA I now call a BioBrick. And this BioBrick I transfer to another bacteria, a host, to give him a new property. I've created a modified bacterium. This technique is of great importance in the field of synthetic biology. Synthetic biology is a new area of biological research. And in this science, we are trying to develop new biological functions for the designing of organisms that are not found in nature. We can do this by engineering known cells and adding new properties to them, but some synthetic biologists are also trying to build completely new cells from scratch. This research is still very new and is not fully developed, but who knows what the future may hold. Synthetic biology will make it possible to design all kinds of different biological systems, but it would be a lot of work to construct all DNA parts separately. It would be far more easy if we could think of DNA standards, like we developed 100 years ago with the screws and bolts. Most of us have a box in the garage like this. We can organize all of our screws and bolts very well. So, for example, if I need a certain part of bolt, I can just open up a drawer, and here I have it. I can start building. Wouldn't it be easy to have something like this with a box full of standard DNA parts? It would look something like this. For example, if I want to have a DNA part that codes for the blue pigment of for the color of my eyes, I would just open a drawer just the same as I did before, and here I have it. I can start building as well. In nature, everything isn't standard. And before I have a large amount of standard DNA parts, it will take some time. This is exactly why the competition iGEM was founded. And my lovely colleague Eva will tell you everything about that.